In 2020, a paper published in Cell Genomics did something no African genetics team had done before. They sequenced the DNA of 400 Fulani people across 6 countries, from Senegal to Sudan, and found a gene that shouldn't have been there. Not shouldn't have been there in the sense of a rare mutation popping up by chance. Shouldn't have been there in the sense that this same gene had been tracked for decades across the frozen coasts of Norway, the green dairy pastures of Ireland, the ancient Neolithic settlements of Central Europe. And now, here it was, at surprisingly high frequency, in a population of nomadic cattle herders stretching across the Sahel, a population surrounded on all sides by peoples who do not carry it. This is the story of how one variant, one single genetic letter, broke everything we thought we knew about African ancestry and forced us to reimagine a forgotten chapter of human migration that reached deep into the heart of West Africa, thousands of years before any European ship touched the coast. The Fulani people, known as the Fulbe or Peul depending on the region, are one of the most remarkable populations on the African continent. They number somewhere around 40 million people, spread across an astonishing 20 African countries, from Senegal on the Atlantic coast all the way east to Sudan. No other African ethnic group is so widely dispersed across such a vast and varied geographic range. They are, by any measure, the single most widely dispersed pastoralist group in Africa. You can find Fulani communities in Mauritania, in Guinea, in Mali, in Burkina Faso, in Niger, in Nigeria, in Cameroon, in Chad, in the Central African Republic. They speak Fula, or Pular, a language belonging to the Niger-Congo family, Atlantic branch, and the language itself carries the trace of their migrations, with dozens of regional dialects spreading along the same Sahelian corridor where their herds have grazed for centuries. Their traditional life is built around cattle. They are, in the deepest sense of the word, a cattle people. Nomadic, semi-nomadic, moving with the rains, following the grass, guiding their herds across a landscape that most settled agriculturalists consider too harsh, too dry, too marginal. The Fulani have not only survived there, they have built a civilization on the move, a culture defined by mobility, by the deep knowledge of animals, by an entire cosmology centered on the cow. And yet, for all their visibility, for all their cultural distinctiveness, the Fulani have always been something of a genetic enigma. They do not look like their neighbors. They do not speak a language from the same subfamily as their neighbors. Their oral traditions trace their origins to mysterious northern sources, sometimes to the Middle East, sometimes to the land of the pharaohs. Arab chroniclers writing about the Sahel centuries ago remarked on the striking appearance of the Fulani, their lighter skin, their finer features, their distinct social structures. Colonial anthropologists, operating with the crude racial theories of their era, concocted elaborate fantasies about lost tribes of Israel, about wandering white tribes, about the Hamitic hypothesis that insisted any trace of civilization in sub-Saharan Africa must have been introduced by lighter-skinned invaders from the north. The Hamitic hypothesis was racist pseudoscience, built on a foundation of colonial arrogance. But it emerged from a genuine observation. The Fulani really did stand out. Something about their history was genuinely different. The question was what? And for decades, geneticists lacked the tools to answer it properly. That changed in the early 21st century as the revolution in genomics began to sweep through population genetics. For the first time, scientists could read the actual genetic text of living populations, letter by letter, and trace the deep ancestral threads that tied them to the past. The Fulani, given their mystery, became a population of intense interest. Multiple teams of geneticists, working across different countries, began to sample Fulani populations. The first major studies emerged around 2011, when a team led by Sirni and colleagues published a detailed analysis of the genetic structure of Fulani pastoralists. That study was a revelation. It confirmed that the Fulani were not simply a subset of their West African neighbors. They were genetically distinct from surrounding Mande, Wolof, and other groups. The Fulani genome showed a clear signature of what geneticists call admixture, the mixing of two previously separated populations. The data suggested that the Fulani genome was roughly 50 to 70% sub-Saharan African, with the remaining 25 to 45% coming from a North African or Eurasian source. This was not a trivial trace ancestry. This was a massive signal, a major component of their genetic makeup. The Y chromosome data, which traces direct paternal lineages, showed a hybrid pattern. Some Fulani men carried the E1B1A haplogroup, the classic Bantu and West African male lineage, but others carried R1b, a haplogroup deeply associated with Western European populations. The Fulani were, in a literal genetic sense, a bridge population, a living record of an ancient meeting between two very different human worlds. But the most stunning finding, the one that would eventually make headlines and force a complete rethinking of African genetic history, was hiding in a single gene. The gene is called LCT, and it controls the production of lactase, the enzyme that allows mammals to digest milk. In most mammals, including most humans, the lactase gene switches off after weaning. There is no biological reason for an adult mammal to continue producing milk-digesting enzymes, because milk is baby food. Once you are weaned, you eat solid food, and lactase production stops. This is the ancestral state. This is the default. But in some human populations, particularly those with a long history of dairying, a mutation has arisen that keeps the lactase gene switched on into adulthood. This trait is called lactase persistence, and it is one of the most famous examples of recent human evolution, a direct genetic adaptation to a cultural practice. If you can digest milk as an adult, you are a lactase-persistent person, and you carry one of several known mutations in the regulatory region of the LCT gene. Here is where the story takes a sharp and unexpected turn. The mutation that allows adult milk digestion in Northern Europeans, the one carried by the vast majority of lactase-persistent people in Scandinavia, the British Isles, and Germany, is a specific variant called LP13910. The scientific designation is RS4988235. This variant emerged in Europe, probably somewhere in the region of Central Europe, and it swept to high frequency in Northern European populations between 5,000 and 8,000 years ago, driven by the intense selective advantage of being able to digest fresh milk from domesticated cattle. This mutation is the reason your Danish friend can drink a glass of milk without discomfort. It is the reason dairy farming took hold in the cold northern regions of Europe. It is, in the genetic sense, a European adaptation. Now, Africa has its own traditions of cattle pastoralism and its own lactase persistence mutations. The East African variants are well known. In Kenya, in Tanzania, among the Maasai and other pastoralist groups, a different set of mutations in the same regulatory region allows adults to digest milk. These variants, LP13007, G, and LP14009. G evolved independently in East Africa, a classic case of convergent evolution. Different populations, facing the same selective pressure of a dairy-based diet, hit upon different genetic solutions. This is exactly what you would expect: independent evolution in response to similar cultural practices. The East African mutations are distinct from the European one. They are not shared. They are separate inventions. So when the 2020 study led by Priehodová and colleagues published their findings in Cell Genomics, sequencing the DNA of 400 Fulani individuals across 6 countries, the scientific community was braced for something, but not for this. The Fulani, these West African cattle herders, carry the lactase persistence variant, but they do not carry the East African variant. They do not carry a uniquely West African variant. They carry LP13910. The European one, the exact same mutation, and they carry it at a frequency of 25 to 40%, depending on the specific Fulani population sampled. This is not a low level. This is a major, functional genetic trait present in a substantial fraction of the population. The neighboring Mande, the Wolof, the Hausa, and every other West African group tested essentially do not carry this variant at all. It is absent. It is a Fulani-specific genetic signature. The so-called European gene no other African population carries is genuinely present in the Fulani, and genuinely absent in every non-Fulani neighboring West African population. This is one of the strongest published examples of pre-colonial Eurasian genetic contact deep into West Africa, and the implications are staggering. Let that settle for a moment. A gene that allows adults to digest milk, a gene that rose to prominence in the cold pastures of Neolithic Europe, is found in a nomadic cattle-herding people stretching across the Sahel, and it is not found in the people living right next door to them, people who speak related languages, people who occupy the same geographic regions. The standard narrative of African genetic history, the one that had been taught in textbooks for decades, held that sub-Saharan Africa was largely isolated from Eurasian genetic contact, until the era of the trans-Saharan slave trade and European colonialism. There were some acknowledged exceptions. The East African coast had contact with Arab and Persian traders, reflected in the Swahili genetic profile. North Africa had continuous genetic exchange with the Mediterranean world. But West Africa, the interior, the Sahel, was supposed to have been a world apart, genetically insulated by the vast barrier of the Sahara Desert. The Fulani LP13910 T variant blows a hole in that narrative. It forces us to reckon with a forgotten migration, a movement of people carrying this gene, a movement that reached the heart of West Africa thousands of years ago, long before any written history records the event. How did this happen? The scientists who published these findings, including the team behind the Vicente 2019 study published in BMC Genomics, titled Population History and Genetic Adaptation of the Fulani Nomads, proposed two main scenarios. The first scenario is that the Fulani share an ancient ancestry with a Proto-Eurasian dairy population, a population that split off in the Middle East and carried the LP13910 T variant before the European and Fulani branches diverged. In this model, the variant is not a European import into Africa at all. It is a shared inheritance from a common ancestral population that lived somewhere in the Near East, a population that had already domesticated cattle and developed the lactase persistence mutation. One branch of this population moved northwest into Europe, where the variant flourished and became the dominant European type. Another branch moved southwest, crossing the Sinai, following the Nile, or perhaps crossing the Red Sea, and eventually made its way into the Sahel, where it mixed with local West African populations and gave rise to the Fulani. In this scenario, the Fulani are not carrying a European gene. They are carrying a Proto-Eurasian gene that Europeans also happen to carry, a relic of a shared ancestral dairy culture that predates the European-African divergence. The second scenario, which is not mutually exclusive with the first, involves a selection sweep from the same Neolithic wave that reached Europe from the Middle East. In this model, the mutation originated in the Fertile Crescent, the heartland of early cattle domestication, and was carried by migrating pastoralists in multiple directions. Some went north into Anatolia and the Balkans, eventually spreading across Europe. Others went south into the Arabian Peninsula, and from there, across the Red Sea, or through the Sinai, into northeast Africa. These Arabian pastoralists, moving with their herds, penetrated the eastern African Sahel, and through a process of gene flow and admixture with local populations, the LP13910 T variant was introduced into the ancestral Fulani gene pool. Once present, the variant would have been under intense positive selection. In a population that depended on cattle milk for survival, especially during the long dry seasons when other food sources were scarce, the ability to digest fresh milk into adulthood would have been a massive survival advantage. Individuals carrying the variant would have been healthier, would have produced more surviving children, and over generations, the frequency of the variant would have risen rapidly. This is the classic signature of a selective sweep, and the Fulani genome shows exactly that signature around the LCT gene. The Priehodová 2020 study went further by attempting to date the inflow of this European-like genetic signal into the Fulani population. The dating is complex, relying on sophisticated models of admixture timing, but the results pointed to a surprisingly ancient timeline. The gene flow started roughly around 1800 BCE, nearly 4,000 years ago, and continued in waves. This is not a recent event. This is not medieval. This is not the Roman era. This is the Bronze Age, a time when the Sahara was already drying, when pastoralists were being pushed southward by the expanding desert, when the great Neolithic cattle cultures of the Sahara were fragmenting and dispersing. The timing aligns with a period of profound climate change and human migration across the African continent. The Sahara had not always been a desert. During the African humid period, which lasted from roughly 14,000 years ago to about 5,000 years ago, the Sahara was a green savanna, dotted with lakes, crossed by rivers, teeming with wildlife. Cattle herders flourished across this vast green space, and rock art across the Sahara depicts cattle, herders, and scenes of pastoral life. As the climate dried and the desert expanded, these pastoralists were forced to move, some southward into the Sahel, some eastward toward the Nile. The Fulani may be the descendants of these Saharan cattle herders, and the LP13910 T variant may have been introduced through contact with migrating pastoralists from the Near East who were part of this broader Saharan pastoral world. The camel is not native to sub-Saharan Africa, but the Fulani mastered zebu cattle, the humped cattle of South Asian and Arabian origin, brought to Africa via Arabian pastoralist migrations. The zebu is not an indigenous African breed. It arrived from Asia, carried by traders and herders across the Arabian Sea and the Red Sea, and it spread through East Africa and into the Sahel. The presence of zebu cattle in Fulani herds is another piece of the puzzle, a cultural and economic marker of the same ancient connections that the genetics reveal. The Fulani are not just herders of any cattle. They are herders of a specific cattle lineage that traces back to the Near East, and their genetic adaptation to milk digestion is tied to the same Near Eastern genetic legacy. This is not a story of replacement. The Fulani are not simply Europeans who moved south. The genetics are clear on this point. The Fulani genome is predominantly African. The sub-Saharan African component is the majority. The Eurasian component, while significant, is a minority admixture. What happened was not a colonization or a conquest. It was a gradual process of mixing, of gene flow, of cultural exchange between herding peoples moving across the porous borders of the ancient Sahara. The Fulani are a genuinely African people, with a deep African ancestry that connects them to the indigenous populations of the Sahel and West Africa. But they also carry a genetic signal of a connection that most other West African populations lack, a connection to an ancient pastoralist world that stretched from the Middle East across North Africa and into the heart of the continent. The Y chromosome data tells this story in stark relief. The presence of R1b, the classic Western European male lineage in Fulani men, is a puzzle that has generated enormous debate. R1b is the most common Y chromosome haplogroup in Western Europe. It is the lineage of the majority of men in Ireland, in Britain, in France, in Spain. Its presence in the Sahel, in a population of African cattle herders, is not something that any simple model of African history can explain. Some early researchers speculated about Roman soldiers, about lost crusaders, about European traders making their way into the interior, but the dating kills those theories. The R1b lineages in the Fulani are not recent European introductions. They are ancient, diverging from the European R1b lineages thousands of years ago. They are not the result of modern colonialism. They are a relic of the same ancient gene flow that brought the lactase persistence variant. The men who carried R1b into the ancestral Fulani population were not Europeans in any modern sense. They were part of a pastoralist diaspora that predated the formation of modern European and African populations. They were members of a shared human story that the neat categories of continents and races cannot capture. The Priehodová 2017 study, published in the American Journal of Physical Anthropology, examined Sahelian pastoralism from the perspective of lactase persistence variants. And it revealed something else that deepens the mystery. The Fulani are not the only Sahelian pastoralists. There are other groups, like the Tuareg, like the Arabic-speaking pastoralists of Chad and Sudan. But the Fulani are unique in their combination of the European lactase persistence variant and their high level of West African ancestry. Other Sahelian groups carry the variant at lower frequencies, or they carry different variants. The Fulani stand out. They are a genetic island of lactase persistence in a sea of lactose intolerance. The selective pressure must have been immense. The advantage of being able to digest milk in a pastoralist economy is not just about nutrition, it is about survival. Fresh milk is a source of clean fluid in an environment where water is often scarce and contaminated. It is a source of calories, of protein, of fat, of calcium. It is a buffer against famine. In the dry season, when crops fail and grain stores run low, the cattle continue to produce milk. The people who can digest that milk live. The people who cannot sicken and die. Over centuries, over millennia, that differential survival shapes the genetic landscape. The Fulani are a testimony to the power of natural selection, acting on a cultural trait, reshaping the human genome in response to a way of life. The broader implications of this discovery are still being worked out. The Vicente 2019 study, the one published in BMC Genomics, did more than just confirm the presence of the European lactase persistence variant. It reconstructed the entire population history of the Fulani, using genome-wide data from multiple Fulani populations across the Sahel. The study confirmed that the Fulani are not a homogeneous group. There is substructure. Different Fulani populations have different proportions of Eurasian admixture. Some are more admixed, some are less. But the Eurasian signal is present in all of them. It is a defining feature of Fulani genetic identity. The study also found evidence of selection on other genes related to immune function, to height, to metabolism, all adaptations to a pastoralist lifestyle in a challenging environment. The Fulani genome is a record of adaptation, a story written in DNA of a people who have navigated the harsh landscapes of the Sahel for millennia. What makes this story so compelling is that it overturns a deeply entrenched assumption about African history. The assumption was that Africa was a continent of isolation, a place where populations stayed put, where genetic boundaries were old and fixed. The Fulani, with their European gene, their Eurasian admixture, their ancient connections to the pastoralist worlds of the Near East, tell a different story. They tell a story of movement, of mixing, of a continent that was always connected to the wider human world. The Sahara was not an impenetrable barrier. It was a highway, a vast space of movement and exchange, and the Fulani are the living proof of that ancient connectivity. The so-called Hamitic hypothesis was wrong, but it was wrong in a specific way. It was wrong because it imagined a one-way traffic of superior civilizers bringing light to a dark continent. The reality is far more interesting. The reality is a complex, multidirectional web of gene flow, cultural exchange, and adaptation, stretching across thousands of years and thousands of miles, connecting the cattle herders of the Sahel to the dairy farmers of Northern Europe through a shared genetic legacy that neither population knew about until the 21st century. The story is not finished. New studies are being planned. Ancient DNA from the Sahel, from the Sahara, from the archaeological sites that dot the old green corridors of the desert, is the next frontier. If scientists can extract DNA from the bones of the ancient Saharan pastoralists, they can directly test the models. They can see whether the LP13910 T variant was already present in the region 4,000 years ago. They can trace the exact routes of gene flow. They can see the faces of the people who carried this gene southward and mixed with the ancestors of the Fulani. The ancient DNA revolution is just beginning to reach Africa, and the Fulani story is going to be one of its most fascinating chapters. For now, the published science stands as a landmark. The Fulani carry a gene that no other African population carries at significant frequency. That gene is the same gene that allows half of Europe to drink milk. It reached the Fulani ancestors through a process of ancient migration and admixture that began nearly 4,000 years ago, during the Bronze Age, when the world was already in motion, when pastoralists were moving across the drying Sahara, when cattle were spreading southward, when the genetic landscape of Africa was being reshaped by forces that historians are only now beginning to understand. The Fulani are not a mystery anymore. They are a revelation. They are a window into a forgotten chapter of human history, a chapter written in DNA, preserved in the bodies of the people who still follow their herds across the Sahel, living the same pastoral life that their ancestors lived when the Sahara was green and the world was young. That's the story so far. The science is settled. The old assumptions are gone. If you want more breaking DNA discoveries, subscribe. Thank you for watching. See you in the next one. Thanks for staying until the very end. Goodbye for now.